Welcome back, biology enthusiasts. Today we're going to continue on with our cell discussion, and we're going to be talking about the cell structures. So starting with any cell, obviously, we're going to talk about the cell membranes. And I'm not sure if you realize this, but not only do cell membranes go around the entire cell, but they go around the different organelles within the cell as well. So that the structures, the organelles have the same cell membrane structure as does the entire cell. So what's the job of the membrane? Probably the most obvious is that it surrounds the cell. It's making compartments within the cell, and it's keeping structures separate from their environment. So your entire cell will be kept separate from its external fluids, and then within the cell, each structure is going to be kept separate from the cytosol or cytoplasm. The other thing that the cell membrane does, I think you might remember a little bit about diffusion. So it controls what can go into and out of the cell, and sometimes what's going in and out has to do with nutrition and waste products, and other times it has to do with communication and secretory products that are going to be used elsewhere. So continuing on with the cell membrane, let's look at its structure a little bit. The basic structure is that it's a phospholipid bilayer. So remember a phospholipid, this is one right here. It has a phosphate group connected to a glycerol, connected to two fatty acids. So similar to a triglyceride, only you're replacing one of the fatty acids basically with a phosphate group. And then that phosphate group gives it a hydrophilic personality, if you will. So this portion here is going to be attracted to fluids. So you'll find it orientating itself towards the outside of the cell because the cell is bathed in fluids, and you'll find it orientated inward as well because the cytoplasm is a lot of fluid, right? Then these tails, because they're nonpolar, they will be orientated to the inside of the membrane, not the inside of the cell, but the inside of the membrane. So these are going to face each other within that membrane. And you might even remember back when we talked about biochemistry, we talked about how these lipids, when in solution, will fold back upon themselves in mycel. So they make little circles, which probably then folded and have infused into the cell membrane. So a little bit more about that cell membrane. It's not just those phospholipids. There's a whole bunch of things in it, so it's considered a mosaic. So kind of like you've seen those pictures where they have lots of little pieces connected, and that makes a mosaic. And so the cell membrane is a mosaic as well. And then because it's in constant motion and it's flexible, it's considered the fluid mosaic model. So you should be able to describe the fluid mosaic model and explain how the different components function within it. So first of all, I see my heads and tails, I see my phospholipids. But what else do I see? I see 1, 2, 3, 4 rings. Remember, that's characteristic of steroids. This particular one is cholesterol. So cholesterol will be packed within these phospholipids, and it keeps those phospholipid tails from sticking together. And this is what allows it to be fluid-like. I see a number of proteins. Some go all the way through the membrane. They're transmembrane proteins. I see some that maybe are just on the outside of the membrane, in which case they're considered peripheral. They can be inside the membrane as well, or towards the inside of the cell. And if they're only on the surface, they would be peripheral as well. And then you have integral proteins, which are amphipathic. They extend into the membrane. Here you see a glycoprotein, so you see the carbohydrate that is attached to it. Remember, we said those serve for cell recognition. So what are some things those proteins do? Down here at the bottom, I have like graphics of each of them, but we're probably most familiar with the channel proteins and the transport proteins. So channel proteins simply allow things to move through like this. They're going to be open. They might be a gated channel, in which case they're closed some of the times, and then depending on the type of gate they have, a ligand will bond and then they open, and then whatever that channel is for will be able to move through. Transport proteins might actually kind of connect with a molecule and move it inward or outward. So if you think about the sodium potassium pump, that would be a transport protein. They are used for cell recognition. I already talked about the glycoprotein. Adhesion. So here you can see some anchoring with microfilaments on the inside of the cell. They can anchor one cell to another cell. That's not it. And so they're holding cells together, intracellular junctions. They are receptor proteins. They receive signals and then pass it inward. That would be signal transduction. And then we're really familiar with cytochrome C, though you haven't been given that name yet maybe, but we're familiar how with the proteins that are in a cell membrane for cellular respiration or photosynthesis, how they act as electron carriers or transfers. So they're moving the electron through the membrane. We also have several proteins within our cells that act as enzymes. So they're functioning like ATP synthase to make ATP. So that's a lot about the cell membrane. You should know its structure. You should understand how the different components within that structure work. And then we're going to talk about the nucleus and nucleolus. And again, both of these have that cell membrane on the outside. It has the same structure. So it's semi-permeable. It has the phospholipid bilayer that protects it from the outside and kind of controls what goes into and out of the cell. So if something is big enough, it's not going to enter the nucleus, or it's not going to leave the nucleus. And maybe that's one of the reasons that DNA can't get out of the nucleus. That's what I like to think. It's double-stranded, it's too big, whereas single-stranded RNA can fit through. The nucleolus is within the nucleus, like small nucleus. And this is where we're going to make the ribosomes. And remember, ribosomes are important for protein synthesis. So within the nucleus, you have your DNA, you have your genetic material. It comes in the form of chromatin or chromosomes. The more visible form under the microscope is the chromosomes. Associated with the endoplasmic reticulum, let's go to that one. The endoplasmic reticulum kind of stretches from the nucleus all the way out to the cell membrane. You have two portions, the rough ER and the smooth ER, so I'm using ER for short. Endoplasmic reticulum. So what makes this rough is that it's studded with these little dots. And what are those little dots? They are the ribosomes. And the job of the ribosome is protein synthesis. So often this rough ER is associated with protein synthesis as well. Ribosomes may be free-floating along the cell's interior as well, but often it's associated with the ER. So the job of the ER is to create glycoproteins. That's one of the things it does. Again, those are going to go to the surface of the cell and serve in recognition. And then you have this portion over here, the smooth ER. This all looks kind of like coral to me in the drawings. So what do we do with the smooth ER? We don't pay a lot of attention to it in 9th grade, but it's for lipid and hormone synthesis. Also, it breaks down toxic chemicals, so you would expect to find a lot of smooth ER in cells of the liver. So continuing on, structures similar to the ER is the Golgi apparatus. So it looks like a bunch of stacks of baggies flattened. They often are referred to as looking like a stack of flattened pancakes. So the Golgi apparatus, its job is to modify and package proteins. So probably the ribosome makes proteins, the ER transports it, and then the Golgi apparatus packages it up for export. So if something's being exported and it has a function, it's called a secretion. Often we use excretion to refer to things that are packaged for waste. So over here, I'm going to talk about the lysosome. The lysosome is one of those little vacuoles that the Golgi apparatus will create, but this has some enzymes in it called lysozymes, and those enzymes, lys means to break up, destroy, and zome is body, so this is a breaking or destroying body. This is digestion for the cell. So this little enzyme disposal bag is going to break up product, and it's going to take it to the surface of the cell, and then through exocytosis, it's going to release its contents outward. So that would be excretion if something's not needed. Over here, the Golgi apparatus also packages things that the cell's going to need. Maybe it's creating a hormone that needs to go from the thyroid gland to another location. So we're going to package those hormones for secretion. And again, that goes to the cell's membrane, and then it opens up as it fuses with the cell membrane because it's made of the same material, that same phospholipid bilayer, and it's going to release its contents outward. Another structure is called the peroxisome. A peroxisome looks just like any other vacuole, I think. So to throw a picture of it up there by itself, you wouldn't know if it was a lysosome or a vacuole or what. So I'm just going to tell you what it does is that it breaks down hydrogen peroxide. So hydrogen peroxide forms when water binds with oxygen. And that can be toxic to us. So we need a way of breaking it down. So it's going to break it back into water and oxygen. And this is common in the liver and in the kidneys and in photosynthesizing plants. So we're going to work with hydrogen peroxide in the lab, and we're going to break it down using the enzyme catalase. Now, how did these structures come to be? Nobody was here, so we don't really know. But the best explanation that scientists have currently is the endosymbiotic theory. So we're going to take two structures in particular, the mitochondria and the chloroplast, and explain how they most likely came to be inside of a cell. So the idea is that they were once individual bacteria. So the chloroplast was a photosynthesizing bacteria that produced oxygen that then gave us our atmosphere. The mitochondria was an aerobic bacteria. So once we started having oxygen in the atmosphere, it was advantageous for this bacteria to be able to use the oxygen to make energy. Okay, so the idea is that they were engulfed by another bacteria and lived symbiotically. So the chloroplast is making food for the host bacteria. The mitochondria is using the oxygen that's created and giving the energy to the host bacteria as well. At the same time, these two structures, the mitochondria and the chloroplast, are being protected from their external environment, so they're going to benefit as well. So with all scientific claims, we should be able to back them up with evidence. So what is the evidence for this endosymbiotic theory? So over here I have the prokaryotes, those are the bacteria, and we have the eukaryotes, that's cells like our own, and then we have the mitochondria and the chloroplasts that are found within eukaryotic cells. So let's look at how these two structures are similar to a prokaryote. So if they're similar, it makes sense that perhaps once they were themselves a prokaryote. So we can see that the DNA of the mitochondria and of the photosynthetic chloroplasts are both single-stranded and circular, just like the prokaryotes, and unlike our own. Did you know that the mitochondria within your cells have their very own DNA? That's kind of crazy. You know what else is cool? Is the fact that mitochondria and chloroplasts, they replicate all by themselves, separate from your very own cells. So that makes them seem like they're not our own cells, doesn't it? So they also replicate the same way bacteria do, through binary fission. Okay, we talked about ribosomes and ribosomal structures in our last lecture. We're making connections! Okay, so look at here, 70S subunits. Well, the whole thing is the 70S ribosome, just like the bacteria and unlike our own. So more and more, these look like little bacteria living inside of our cells. Again, electron transport chain. So we'll talk about this with photosynthesis and cellular respiration. The electron transport chain is found in the membrane of these cells as well as in the membrane of the entire bacteria. Not true of our own cells. They're only found within the mitochondria or the of the chloroplast, sorry. And then we can continue on to the sizes, similar. They're both little tiny things like bacteria. The only thing that doesn't correlate is the time frame. So we figure these anaerobic bacteria were 3.8 billion years ago. So anaerobic, they didn't use oxygen. There was no oxygen in the atmosphere for them to use, so they didn't need to. And then we had these photosynthetic bacteria starting to develop about 3.2 billion years ago. So what were they doing photosynthesizing? They were making oxygen. So now doesn't it seem reasonable that a mutation along the way that allowed some bacteria to be aerobic would be advantageous, because now they can use the oxygen that's in the atmosphere. So that's the background of these bacteria, and then all these other cells and started coming around 1.5 billion years ago from the best of our ability to date them. So you should be able to identify the evidence behind the claim. So why is it that we're allowed to say mitochondria and chloroplast most likely were once bacteria and they were engulfed by another bacteria and they live symbiotically? So now you have some evidence that you can use to support that claim. So this is just a graphic representation of how endosymbiotic theory might have worked. So here you have the host cell, here's a host cell, you have your aerobic bacteria, you have your photosynthetic bacteria, and this is that process of phagocytosis. So the membrane will surround a structure or being outside of the cell until the two pieces come together and meet, and then they will fuse together, and then when that happens, this portion will drop down inside as a vacuole, and in this case, it remained on the inside as the mitochondria or as the chloroplast. So another structure that is common to many cells is cilia and flagella. So some of your cells have cilia on them, like your respiratory tract has cilia, your reproductive tract has cilia. Many protozoans have cilia, so maybe these little tiny hairs are moving to allow the whole organism to swim, to move, or maybe these little organisms are sweeping food into a mouth portion. Flagella are usually longer, similar structure, and they're always used for propelling the organism. So this is some bacteria that has flagella on it. So they're both made of microtubules or microfilaments arranged in a 9x2 arrangement. So if I counted these up around the circle here, there's 9, and they come in sets of 2. So that's a common characteristic of the cilia and the flagella. So I tried to pick some of the most common structures that we'll talk about. We talked about the ribosomes, the nucleus. We did not talk about the centriole. That appears around the time of cell reproduction. So we'll talk about that more when we talk about mitosis. Here we have microfilaments that might be part of the cytoskeleton as a whole, or maybe they're used in creating spindle fibers or flagella or cilia. We talked about the smooth detoxifying endoplasmic reticulum, the mitochondria, which makes energy. The rough ER, which is associated with ribosomes and is used for protein synthesis when it's associated with ribosomes, as well as glycoproteins, and the Golgi apparatus that is going to package proteins for export, secretion, or it's going to package up waste products with enzymes that will allow it to break down. So that's all I have for you today. Thanks for listening.